I will present the interim data from our first in-human study of an in vitro CRISPR Cas9 based gene editing therapy, NTLA-2001, which is designed to edit the TTR gene in patients with transthyretin amyloidosis. I'm going to focus very much on that one trial. Obviously, I'm very aware that many of you have been involved in all sorts of other bits and bobs and other trials in patients of this nature, but I'm going to focus on this one particular trial. My disclosures. So just to start off, amyloid is a misfolded fibrillar protein deposit in the tissues, which is characterized by pathognomonic green birefringence when you stain that tissue with Congo red, the dye Congo red, and view it under cross-polarized light. And there are about 30 different proteins in vivo in the human body that can misfold and form amyloid fibrils in vivo. And they form the basis for the classification of the different types of amyloid. Okay, so many of you will have heard of AL amyloidosis, AA amyloidosis. All types of amyloid start with A, and the second letter stands for whichever protein it is that makes the amyloid deposits. So in AL amyloidosis, it's a light chain. In AA amyloidosis, it's a protein called amyloid A protein. And in ATTR amyloidosis, the condition that we're talking about today, the protein that makes the amyloid fibrils is transthyretin, or TTR for short. So in ATTR amyloidosis, the amyloid fibril protein is derived from misfolded TTR, as I mentioned, and it can be either acquired or hereditary. The acquired form of ATTR amyloidosis is known as wild-type ATTR amyloidosis, and what we know now is that it is probably a not uncommon cause of heart failure in elderly individuals. And in our cohort, about 94% of patients are male. We don't understand why. And one incredible fact is that if you look in post-mortem specimens in individuals over the age of 80, 25% of them contain ATTR amyloid deposits in their hearts. Okay, now we certainly don't diagnose 25% of the population over 80 with cardiac amyloidosis or cardiac ATTR amyloidosis, but what we do know is that a lot of those patients never get diagnosed. The vast majority are never diagnosed in life. But what we also know is that a significant proportion of those patients almost certainly have ATTR amyloid deposits in their hearts that are not causing a clinical syndrome. But we don't know where along that spectrum, are we missing millions of patients with ATTR amyloidosis who have a disease and are never getting diagnosed? Or are the vast majority of those individuals just have ATTR amyloid deposits in their hearts but don't actually have a disease? We don't know where along that spectrum we are. But what we do, so consequently we don't know the prevalence, but you can see in that graph on the right there the increasing number of diagnoses of ATTR amyloidosis that we have had in recent years, down to better imaging techniques and greater awareness amongst cardiologists. Hereditary ATTR amyloidosis is associated with more than 130 mutations in the TTR gene. It's inherited in an autosomal dominant fashion, and it can be very, very similar to the wild-type disease, which is a predominant cardiomyopathy, although quite often it affects the nerves as well as the heart. And many of you have experienced some patients with neuropathy and cardiomyopathy. The variants that we see most commonly in the UK is the V122I variant, which occurs, in fact, the mutation occurs in 4% of people of African descent. And that gives rise to a clinical syndrome which is very similar to the wild-type disease, so a predominant cardiomyopathy. And the other variant that we see in the UK is what we call the Irish variant, if you like. It's T68, and that gives what we call a mixed phenotype with neuropathy and cardiomyopathy. And some patients, even in the same family, will have more severe neuropathy another member of the same family with the disease will have more severe cardiomyopathy. But most patients will have involvement of both of those organs. So the main therapeutic strategy for treating every type of amyloid that we've ever treated is to suppress the concentration of the amyloid forming protein. So in this case, it's transthyretin, okay? And Jörg touched on gene silencing therapies, and we've got 2 gene silencing therapies that are approved in the NHS, inotersen and patisiran. And essentially, they lower TTR levels in the bloodstream by about 80%. And they've been shown in trials to arrest the progression in many patients with hereditary ATTR amyloidosis. And in some patients, there are improvements in neuropathy scores. But it's certainly, that is not necessarily the norm, if you like. Editing the TTR gene is a pretty attractive possibility in that it has the potential, of course, to provide permanent and profound TTR knockdown of greater than 80% after a single infusion. The gene editing treatment, certainly patisiran, probably the more effective gene editing treatment, needs to be given by intravenous infusion every 3 weeks for the rest of a patient's life. And there are pre-meds associated, so it's certainly not particularly convenient. So I'd just like to use this slide, hopefully, to convince you that the more you knock down the relevant protein, the better the outcome. Okay, and on the left there, on the left you can see this is AL amyloidosis, and the relevant protein is the light chain that I was talking about. That is the protein that makes the amyloid fibrils. And what this shows is that if you achieve what we call a complete clonal response with chemotherapy, in other words, you knock down the light chain by 100%, then patients do very well. If you fail to knock it down, that's no response at the bottom there. And in between, you have what hematologists call a very good partial response, in other words, a 90% knockdown of the relevant protein, and here a partial response, 50% response. Okay, so a direct relationship between the degree to which you knock down the protein and clinical outcome. And exactly the same is true in AA amyloidosis. I told you that the protein that makes the amyloid in AA amyloidosis is a protein called SAA, or serum amyloid A protein. Okay, that's an acute phase protein. It's exactly like CRP. It goes up when any patient has a high CRP in their bloodstream, they have a high SAA. It's just that we don't measure it. And if you treat their underlying disease, bring down their SAA, the more you bring it down, the better the outcome. You can see a direct relationship here between SAA concentration, octiles of SAA concentration, and clinical outcomes. And what's notable here is that even the normal concentration is less than 4 milligrams per liter. And the relative risk of death between less than 4 and 10, which is still a pretty low level of SAA, albeit not quite normal, is nearly 4 times. So evidence that the more you knock it down, the better you do. And you can see that across the spectrum. The same data in ATTR amyloidosis is actually not there yet. And the reality is that we don't know whether this will stand up in ATTR amyloidosis, but I would eat my hat if it doesn't, is the honest truth. Every type of amyloid, I've shown you a couple of examples, but every type of amyloid that's ever been treated, the more you knock down the protein, the better patients do. So, there is absolutely no reason that it should be any different in this particular type of amyloidosis. So, NTLA-2001 is a CRISPR Cas9 based in vivo gene editing therapy. It consists of a single guide RNA that is targeted to the TTR gene and an mRNA, human-optimized mRNA, the sequence of strep pyogenes, encapsulated in this lipid nanoparticle. Okay? When you administer NTLA-2001 intravenously, it's transported directly to the liver and taken up by the liver via the LDL receptor on the surface of hepatocytes. It's endocytosed and released into the cytoplasm, and the Cas9 mRNA is translated, producing the Cas9 enzyme, and that interacts with the guide RNA to produce what we call a ribonucleoprotein complex, CRISPR-Cas9 RNP complex. That then is transported to the liver, and a 20 nucleotide sequence on the guide RNA, on the 5' end of the guide RNA, then binds to the DNA double helix at the target site, in other words, at the TTR gene, and cleaves the TTR gene. Okay? And as a result of that, you don't get mRNA production and therefore don't get protein production. So that's the principle, if you like, of the treatment. So this plot shows you the in vitro therapeutic index with effects of concentration of the guide RNA on the percentage TTR gene editing by NTLA-2001 in cultured primary human hepatocytes. The vertical line here is the EC90, which is the concentration at which one regularly sees at least 90% reduction in TTR protein concentration. Okay? And what you see is above the EC90, there's a saturable level of TTR gene editing. Okay. One really important question, and this is key to this CRISPR-Cas9 technology, one really important question that the scientific team at Intellia asked is, what is the rate of what we call off-target editing? That's the big risk of CRISPR-Cas9, okay? What you want to do is edit the gene that you're targeting, but you don't want to start editing genes that you inadvertently happen to chop off or edit without meaning to. And that's what we call off-target editing. And the Intellia team performed a number of studies, some computational analyses, some in vitro assays, cell-free, cellular-based assays, and they basically identified 7 sites that are, that they consider at risk of off-target editing. Okay? Now, all of those sites were in non-coding regions of the genome, and that's important. And what this figure shows you, if you like, is the relationship between on-target editing, in other words, editing of the TTR gene, versus editing of these 7 sites. And the important thing here is that at concentrations of up to 3 times that of the EC90, you have no detectable off-target editing. So I think that's a really, really important point. And that's probably an important advance that is, that Intellia really are responsible for, which has enabled this to be potentially at least given safely. So the preclinical in vivo study was conducted in the cynomolgus monkey using a surrogate LNP that is obviously specific for the monkey TTR gene. And what this graph shows is the percentage reduction from baseline in serum TTR protein concentration in monkeys receiving the LNP at these various different doses. And what you can see is that a dose of 3 milligrams per liter led to 95% reduction from day 28 after a single infusion, and essentially the monkeys were followed for 367 days. Safety studies established obviously a dose range at which no adverse events or effects of NTLA-2001 were observed, with near complete sustained reduction in serum TTR following a single administration. At higher doses, the adverse effects in monkeys were dose dependent, they were transient, they were reversible and monitorable. So the first in-human study with NTLA-2001 is a 2-part open-label multicenter study in patients with hereditary ATTR amyloidosis and polyneuropathy. It has, as many of you will know, just been amended to include patients with cardiomyopathy and patients with wild-type ATTR amyloid cardiomyopathy. So part 1 is a single ascending dose design with a minimum of 3 patients in each of up to 4 dose ascending cohorts. And the starting dose in humans was 1/10 of the allometrically scaled dose at which no adverse effects were identified in the monkey. And then the recommended dose from part 1 will be administered to a dose expansion cohort in part 2. The primary objective, obviously, of this first-in-human study is safety, tolerability, PK, and PD. PD is by serial measurement of TTR levels, which is a surrogate of likely clinical outcome. So I'll report data. I'm only at liberty, I'm afraid, to report data on the first 6 patients from those first 2 dose escalation cohorts in part 1. Bear in mind, these patients obviously received a single dose on day 1 of the study. These are the demographics of the patients. First 6 patients, 3 in each cohort. Age, as you can see, ranged from 46 to 64. There were 3 different pathogenic TTR mutations, and 3 patients had received a prior TTR stabilizer drug as a potential disease-modifying therapy, but they stopped it prior to being given this infusion. All had basically early-stage polyneuropathy, and although 5 out of the 6 had evidence of cardiac involvement by imaging techniques, they were all NYHA class 1. Broadly, the bottom line is that these patients all had early-stage disease. NTLA-2001 was generally well tolerated in the acute phase, with AEs all being of mild or grade 1 severity and mostly unrelated to the treatment. There was a patient who had an infusion-related reaction, as you will know here better than I do, so I'm embarrassed even to tell you about that, but I gather it didn't require dose interruption. And there were no SAEs in these first 2 dose cohorts. There was no evidence of liver toxicity or coagulopathy, and all the blood tests were essentially, the laboratory excursions were clinically insignificant. And I suppose this is this is the exciting data. So this is the serum TTR reduction, which you can see drops by day 7 and deepens further by day 28 in all patients. And at the first dose level of 0.1 milligram per kilogram, the reductions range from 47 to 56%. And as you can see here, in the reductions were dose dependent, with greater reductions detected in 3 patients who received the higher dose of 0.3 milligram per kilogram. And here the reductions range from 80 to 96%. We've never seen 96% reduction in TTR with any other treatment over the course of many, many years. At day 28, the mean TTR reduction from baseline in the 0.1 mg per kilogram cohort was 52%, and in the 0.3 milligram per kilogram cohort was 87%. Pretty impressive. So to conclude, systemic administration of NTLA-2001 in patients with hereditary ATTR amyloidosis and polyneuropathy caused a profound and dose-dependent reduction in serum TTR protein concentration. Of note, the maximum reductions were 56% and 96% in the 2 dose cohorts. NTLA-2001 at these doses was well tolerated in the acute phase, with the AEs being of mild severity. Obviously, as you will know, further dose escalation is planned or has occurred in some cases. And what we expect is that the dose escalation will lead to greater reduction in TTR. And as I told you earlier, I believe that that will lead to greater clinical improvements. I think one important thing to mention is that the prognosis in hereditary ATTR amyloidosis, obviously in the case of wild-type ATTR amyloidosis, it goes without saying, but the prognosis is really driven by the cardiomyopathy, by the heart disease. And on the basis of the data that I showed you in AL amyloidosis, it's completely conceivable that the real benefit of this increased knockdown with CRISPR-Cas9 compared to the gene silencing treatments, if you like, will be most apparent in the patients with cardiac involvement. So this is the first demonstration of CRISPR-Cas9 based in vivo gene editing in humans, and obviously provides a proof of concept for a pretty promising therapeutic strategy. I mean, I think the real important thing is that producing your guide RNA is actually very, very straightforward. You can produce your single guide RNA to any gene in the body. And so technically you can edit with this technology. It's modular. You can edit any gene in the body. The challenge is delivery of your drug containing your single guide RNA to the right cells if this is going to change medicine, you know, in terms of the long term. And then finally, it leaves me really to mainly thank you guys, because you did all the hard work. I do the talking, you guys, I get the credit, but you guys do all the hard work. So, you know, really thank you to all of you. Obviously the guys in New Zealand who were the other main center. And then ultimately the clever guys at NTLA who have really developed this technology and, you know, hopefully made it safe for people in vivo. So thanks very much for listening.